continuing on with Gregor Mendel, okay, we can look at the results of his experiments, crossing these various pea plants. We're first going to focus on purple and white flowers. And so he got these true breeding flowers, which essentially means they're homozygous. Anytime you self fertilize them, they always produce flowers of the exact same color. Okay, so we've got our parental generations shown as P. He cross fertilized a white with a purple flower. And in the first filial generation, the F1, he found that all of the flowers produced were purple from these offspring plants. Okay, interesting. Okay. He then took those F1 plants, self fertilized, so he guaranteed that whatever genotype these plants have, they'd be mixing with themselves. Then he planted those seeds, and then he counted up all of the flowers and figured out what color they were in this F2 generation. He found it was about 705 purple to 224 white, or a 3-to-1 ratio. So we see 3 out of every 4 were purple, 1 out of every 4 were white. So by mixing these two purple flowers that he got from mixing purple and white, we end up with 3 purple and 1 white here. Okay, so how do we explain what we see? Okay, he then went and made an F3 generation by self-fertilizing this F2 generation. Okay, all the white flowers produced more white flowers. They were true breeding. Some of the purple produced purple and white, but some of the purple, 1 out of the 3 of them, produced all purple flowers. It was pretty interesting. Okay, and we'll look at some examples with Punnett squares of why this came out to be this way. And what we essentially find is that purple flowers are dominant, white are recessive. So in that first generation, the F1, those traits get covered up. Anyway, what we find in Mendel's experiments is that we've got these distinct units that are passed on from parents to offspring. Okay, and we know this. We've got genes in our chromosomes. It's something we already know. Each individual has 2 alleles of a given gene. Okay, this is information that we've added on since Mendel's work in the 1800s. Okay, and so we can have these 2 different alleles. We could have this dominant recessive relationship, whatever. They could be homozygous dominant, homozygous recessive, heterozygous. And 1 allele of each gene is on each of the homologous chromosomes. So we get 1 of those alleles from mom, 1 of those alleles from dad. So we've got this idea of segregation, 2 copies of a gene, but put only 1 copy in each sperm and egg, so that when fertilization occurs, we get 2 copies. Okay, and remember that law of independent assortment, where the alleles that we have, our chromosomes, are just kind of randomly going into these gametes. So we've got all of these different possible combinations, and then when we get fertilization, it increases even more. So we have Mendel's law of segregation that he came up with. So the pairs of alleles separate from each other during gamete formation. We know this. This is the law of independent assortment. When sperm fertilizes the egg, the offspring gets 1 allele from mom and 1 allele from dad. Okay, so we get these brand new combinations of genes in these new individuals. Alleles end up in gametes by chance. Again, this is independent assortment. These gametes are involved in fertilization by chance, right? For the most part, we're not choosing which gamete is going to be part of fertilization. Okay, eggs kind of get released whenever, and you can't determine which of the 100 million sperm are going to fertilize that egg. Okay. What did Mendel's findings reveal about inheritance? Okay, he found that individuals that were heterozygous or hybrids had 2 different alleles. One of those alleles was dominant, the other was recessive. And so we get a phenotype of the dominant allele because it covers up the recessive one. He found that with homozygous or true breeding plants, both alleles are the same. And so we've got that big P gives us purple, little p gives us white. Right? And so our genotype in the homozygous plants reflect their phenotype. Okay? Looking at Mendel's results, he got this 3-to-1 ratio when he self-fertilized these heterozygous plants. Okay? And we can look at it with this Punnett square. So Punnett squares are tools that allow us to consider the probabilities of certain parents having various offspring. Okay, and again, I've got videos posted of this type of Punnett square practice that you can look at to get an idea of how this is done. Okay, so this is Mendel's F1 generation where he self-fertilizes the heterozygous plants. And so then we just carry on these letters, big P come into this box, big P comes down with a little p because it's heterozygous, big P comes over, little p comes down, then little p. And so this is why we end up with, when crossing these heterozygotes, 3 purple plants and 1 white plant. We only get 1 white because it has to be homozygous recessive in order to have that phenotype. We also get 2 heterozygous plants, and we get a homozygous dominant plant. Okay, so what's the phenotype of this plant? Purple flower, right? We can see that physical expression. What's its genotype? Okay, use that term that we would use when we refer to genotype. Heterozygous in this case. If we let this plant self-fertilize, what would be the expected phenotypic ratio? 3-to-1, right? Just like we saw in our example that Mendel did. Okay, and you can do a Punnett square to figure this out. What's the genotypic ratio? So usually when we look at genotypic ratios, we go number of homozygous dominant, number of heterozygous, number of homozygous recessive. So here we've got 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So our ratio is 1-to-2-to-1. So now you can cross these plants, figure out the phenotypic ratio. So we've got a homozygous dominant and a heterozygous. And so you can work this out, and basically what we're going to see is all of the plants are going to end up purple, because from the homozygous dominant parent, all of the children are going to receive that dominant allele, so they'll all end up being purple. So our ratio is 4-to-0 here. 6, crossing 2 white plants, what's the phenotypic ratio? Well, we know that in order for the flower to be white, it has to be homozygous recessive. If you cross 2 individuals that are homozygous recessive, the only alleles that the offspring can inherit are the recessive alleles. So all of the offspring will be white. And so we're 0-to-4 with this phenotypic ratio. Okay. These were all monohybrid cross Punnett squares where we just look at a single characteristic at a time. We get a nice 2-by-2 box. You can extend this and look at multiple traits at the same time. Okay, so Mendel did experiments looking at seed shape and seed color, whether the seed was smooth or wrinkled, and then yellow or green. So we've got multiple traits here. So we end up with a 4-by-4 Punnett square, 16. And so if we've got 2 sets that are heterozygous, always going to get a ratio of 9-to-3 to 3-to-1 in our different expressions here. Now, I will tell you I'm not going to make you do any dihybrid crosses. You will be expected to do monohybrid crosses, though. Okay. All right, so law of independent assortment. Again, these traits are inherited independently, which is why we get this mixture of smooth and green and smooth and yellow and wrinkled and yellow and wrinkled and green when we look at multiple traits, okay? Because they're on different chromosomes, they're inherited separately. Okay, so we'll continue on from here in the next video.